Right, can we settle down? We're about to start the session. Okay, so good evening, everyone. I'm Kirtan Deda from the University of Cape Town, and I want to welcome you to the Lancet Respiratory Medicine Symposium. This is the second symposium we're having, and the one last year was very successful and was the launch of the Lancet Respiratory Medicine Commission. In fact, if you walk into the exhibition hall on the right-hand side at the Lancet Respiratory Medicine stand, you can actually pick up a copy. And there'll be many other commissions to come that will be published in Lancet Respiratory Medicine, and one of the features of the commission is that every few years the commissions will be updated. Now, the main purpose of this symposium is really to showcase translational research that has clinical impact. It's also an important advocacy platform for respiratory diseases, particularly TB. And TB, as we all know, has a number of non-communicable sequelae. It's arguably the commonest cause of pulmonary disability worldwide, commonest cause of pulmonary fibrosis. I keep reminding Ganesh Raghu from the USA, and a very important cause of COPD. And of course, we mustn't forget TB also spawned the field of respiratory medicine or pulmonology as we know it today, and the Grey Journal and the Blue Journal, in fact, all started off as TB journals. We have a very exciting lineup of talks today by several experts who are leading authorities in their respective fields of interest. And to kick off the session, I'm going to invite Professor Tawanda Gambo from Baylor, who will chair the first part of the session.
So before we start, I just would like to thank you all for attending today. And what we're going to do is the speakers will give their presentations, and then there will be a discussion that will follow later. Our first speaker is Professor Kirtan Deda, who was standing here. He is one of the leading voices and group leader in the field of tuberculosis, especially drug-resistant tuberculosis. He is based at the University of Cape Town in South Africa. He's an avid cyclist. We were just talking about that here. And he is going to give the first, and he will introduce the next speaker after he is done, and we'll keep doing it that way. Kirtan.
So thanks Tawanda for that introduction. So just to get my conflict of interest out of the way, I have received some grant funding and speaker fees from FIND and Cepheid, but it's not directly related to the content of this talk. So to give you a brief overview of my talk, I'm really going to talk about two things. The first is how to prevent emerging resistance to newer drugs, and to give you some insights from our lung explant studies. And the second thing I'm going to talk about is the diagnostic gap, or the more than I think roughly 4.2 or 4.3 million undiagnosed cases or missing patients, and how do we decentralize diagnosis to try and address this problem. So as all of you know, TB is far from eradicated, and the latest global TB report has just been published, and says that in 2015 there were an estimated burden of almost 600,000 MDR TB cases. And as you can see from the graphs that I've put up there from that report, there's an increasing detection rate of MDR TB in many of the high burden countries. The other thing that's remarkable in the report is that the degree of resistance of isolates is increasing. So in contradistinction to the 2014 report, where this figure was about 30%, it's amazing that 51% of rifampicin-resistant TB or MDR TB had resistance to either fluoroquinolone, a second-line injectable agent, or both. And about 20% of strains globally now is resistant to at least one major first or second-line drug. And the reason I put in that picture inset is that it's not only a problem confined to resource-poor settings, it's a major problem in Europe. Almost 80,000 cases MDR cases detected in 2015. And there you can see a queue of migrants, and Germany alone last year had over 1 million migrants. Drug-resistant TB also has a very high mortality, somewhere between 40 and 70%. And we're never going to tackle the problem of TB mortality if we don't tackle drug-resistant TB. And as you can see from this graphs from the BRICS countries, again in the latest TB report, and from data we published in PLOS One, about 40% of total TB funding at the moment is on drug-resistant TB, despite that making up less than 5 to 10% of the total caseload. And we've now progressed to programmatically incurable TB. And this is in the main patients with XDR TB who, despite receiving 12 or 18 months of treatment, remain therapeutically destitute or treatment failures. And because we have no facilities to house these patients in most resource-poor settings, in South Africa, for example, these patients are discharged back into the community. And the same thing happens in India, Russia, and China. And we showed in this paper earlier this year that these discharged patients actually spawn secondary cases of incurable TB. And you may say, well, that was in the pre-bedaquiline era, and now we have bedaquiline, and so that solves the problem. But if you actually look at the aggregated data, even with bedaquiline, 24-month XDR failure rate was about 38%. So newer drugs will, of course, markedly reduce treatment failure rates, but it won't eliminate it. And here's just a couple of case reports of patients that are resistant to both bedaquiline and delaminid. And last month alone at our respiratory clinic, we had about 10 patients that were referred for lung resection, having failed both bedaquiline and delaminid. And so people have started asking whether we need to build the modern versions of facilities like this. So the real question is, what is the cause of drug-resistant TB? And I think it's critical to address this question, because if we're going to keep doing what we did so far, we're going to end up rapidly losing the new drugs that we have. So what is the cause of drug-resistant TB? Okay, so I see we don't have any sound. I thought I'll play this video clip, but anyway, we'll skip that. So the traditional thing we did is to blame the patient, right? We said drug-resistant TB is because of poor compliance. Patients didn't take their treatment. They had a lot of psychosocial issues. They never completed their courses of treatment. And we had sequential monotherapy and development of resistance. And however, in fact, that's probably was a violation of patients' human rights, because in fact, despite perfect adherence, we know that perfect adherence does not prevent the emergence of resistance, and there's a number of randomized controlled trials that show that. The real cause of drug resistance is PK mismatch, and by that we mean a suboptimal number or level of a drug relative to MIC, and there are lots of causes of PK mismatch, and I've listed them there. I'm not going to go through them in this slide, but the one neglected area that we don't talk about a lot is drug gradients within TB lesions. This is a paper from Véronique Detroit's group, which used a relative mass spec imaging of slide sections, and you can generate these heat maps, as you see in this figure, where you can look at penetration of drugs into these necrotic or caseous lesions. And the message from this paper is that the suboptimal penetration of drug into these necrotic lesions or cavities. But there are a number of unanswered questions. How does this relate to MIC? And what are the absolute drug levels in different parts of the cavity? So to address this question, we completed a study which has been submitted for publication now, where we looked at 14 patients. These were patients who had failed drug-resistant TB treatment, and the lungs were resected and taken to our lab. And what we did is, in these individual cavities, what we did was we biopsied these cavities. So as you can see in this cartoon, the biopsy point 1 is normal appearing lung tissue adjacent to a cavity. And then sequentially you can see numbers 2 and 3 represents the wall of the cavity, and then 4 the inner wall of the cavity, and 5 the center of the cavity. And in tandem, we took a sputum sample, and we also had sputum samples at the time of diagnosis and serial sputum samples, because this was part of a larger parent study looking at prospective outcomes in patients with drug-resistant TB. And at all these sites, we did whole genome sequencing. We looked at MICs at all these biopsy sites. These were 2 by 2 millimeter biopsies at each of these sites. And we also looked at absolute drug levels. So just to show you the data, firstly, I just want to demonstrate the clonal heterogeneity you get within a cavity. So if you just look at that table on the left-hand side, that's patient 2, and you can see the different points. So for example, point 2, I told you is normal appearing lung tissue, and then point 3. So you can see in this particular patient, in sputum, there was an X1 strain. And at point 2, which is the normal appearing lung tissue adjacent to the cavity, there's X1 strain. And then when you get into the wall of the cavity, you get a completely different strain, right? And that's been demonstrated in a couple of small studies. And that's just a different way of demonstrating the data using these genotyping dendrograms. And each color there represents one patient. And so the further away from the edge of that diagram, these stepped lines go, then the more unrelated these isolates are, basically saying that if you look at a cavity, it's all not one strain. So moving on from there, I'm now going to talk about the MICs at each of these biopsy points. And what you can see from this graph, it's a bit of a busy slide, but each of these lines represents one patient. So for example, if you look at the black line, you can see this patient in the sputum was resistant to, in this case, moxifloxacin. You can see the break point here. But when you look at the airway and the, you can see that it was sensitive, and then moving towards the center of the cavity, resistant, and in the cavity itself, sensitive again. So you can see that the MICs vary quite a bit. These were done on 2 occasions at 2-fold dilutions, and the same picture with a number of other drugs. Then moving on from there, we looked at drug levels. And again, I'm just using MOXIE as an example. And this is aggregated data from the 14 patients. So you can see in blood, there was the level of moxifloxacin and then the wall of the cavity. And you can see when you get into the cavity center, there's a massive decrease in the concentration of drug you find in the center of the cavity, basically mirroring the findings of the paper that I showed you, that there's very poor penetration of some drugs into the center of the cavity. And if you look at what this graph shows you is if you look at the center of the cavity, on average, there was only one drug at concentrations above MIC. And so you can generate heat maps like this, and this just shows you the drug for this example is moxifloxacin again at the various different biopsy points, the cavity center. And what this shows you, it's a heat map of correlation. So essentially, that's a perfect negative correlation being dark blue. And what this is showing is as you move from normal appearing lung tissue into the center of the cavity, you get a darker and darker blue, basically showing that as the MOXIE levels decrease, MIC goes up. And this was in the setting of acquired drug resistance because, as I told you, we had serial sputum samples demonstrating that there was increased acquisition or evolution of resistance in these patients. So the key points from this part of the study was that there's considerable heteroresistance in a cavity, But the main point was that there's a decreasing drug gradient across the cavity wall associated with increasing MICs during the course of development of acquired resistance. And of course, this supports the hypothesis that there are windows of monotherapy that likely drive resistance. And so what can we do? One of the things that we could do is give drugs in an alternative way so that we could use adjunctive inhaled antibiotics. In this case, proof of concept study from Ed Nardell's group, capreomycin. And you can see at the highest dose, 300 milligrams, you get reasonable serum levels. The other interesting thing we found in the study, comparing sputum versus the cavity, what this graph shows you is the predictive accuracy of sputum. So you can see, for example, if you look at moxifloxacin, then what the DST in sputum was only 30% predictive of what we found in the cavity. And if you average it out for all the drugs, then there was only an predictive accuracy of 60%, suggesting that the readouts we get in sputum is far from the reality of what's happening in the cavity. And so maybe, as clinicians, it's not so surprising for us when patients fail therapy, or we find that it's MDR TB, and they're sensitive to second-line injectable drugs and fluoroquinolones, and later we find they've evolved to XDR TB. So maybe that's not so surprising. And so we can use approaches like this. This is deep sequencing, and Megan will talk more about this. But essentially, what we do here is extract DNA, amplify the DNA using using PCR, and then do targeted sequencing. And so you can detect rare populations in sputum. We call that micro-heteroresistance. And perhaps we need techniques and technology like this to tell us what's really happening in these cavities. And so the implications of this is that we need alternative approaches to prevent the amplification of resistance. I spoke about adjunct inhaled antibiotics, therapeutic drug monitoring. We often don't know about whether we're optimally dosing these patients, so therapeutic drug monitoring for optimal dosing, and Tawanda will talk more about that. Use of efflux pump inhibitors, pharmacogenomics, and maybe we need to also consider this when we're selecting drugs for clinical trials, right? So maybe the drugs that we ought to be selecting to move forward into phase 3 trials are those with optimal intracavitary penetration. And maybe we need to think about the way we do diagnosis, and maybe we ought to be doing, besides enabling sequencing directly from the sputum, we need to be developing diagnostic tests that can detect micro-heteroresistance. And this really all speaks to precision medicine or individualized therapy for drug-resistant TB. So in the last few minutes, I just want to talk about gaps and dropouts. So this is a very interesting study published earlier this year from India. And this was referring to 2 million patients with TB that entered the Indian healthcare system. And what you can see is along every step of the way, there's attrition. So there were some people who, in fact, were not diagnosed with TB. Some were diagnosed with TB but never started treatment, and so forth. And the bottom line is, at the end of the day, there were only half the patients that were actually relapse-free at 1 year. So these are people that actually reached a healthcare facility. But there's a whole bunch of cases that never reach healthcare facilities. And this is the diagnostic gap, or the missing cases I was talking about. And you can see in 2013, that number was 3 million. It's gone up to, estimated, gone up to 4.26 million. And of course, this is even worse, these numbers or proportions, for drug-resistant TB. And really what this is all about is that these patients are drivers or drive transmission, because these are undiagnosed cases. In the main, some are unreported cases, but in the main, these are undiagnosed cases in the community. And how do we address this problem? So we published this study. In fact, it was published in print earlier this year. And what we did is we put a gene expert machine on a van, and we went out into this community of Langa in Cape Town. And you can see the sputum induction tent there. And this van was parked at congregate settings and in entrances to informal settlements within this township. And why the study was important. It was the first randomized controlled trial showing the impact of molecular diagnostics for active case finding. And in fact, we've now recruiting for EXACT-2, and this is an NIH-funded study where we're using Expert Omni, and this is in a small. Let's go back. This is in a. This is in a more scalable, small panel van with 2 healthcare workers, and I just wonder in several years whether we might be able to take sequencing, in fact, out into the field. So this is a Nanopore MinION technology, and it's still early days, high error rates, but I think maybe Megan will tell you more about this. But they're also working on a version now that you can attach to a smartphone. This is sequencing attached to a laptop. So maybe in several years we might get this out into the field, as we did with this active case finding approach. So we really need to think about how we're going to diagnose cases, because this is the elephant in the room. And if we're going to continue doing what we always do, we're never going to get on top of TB, and we need to think about innovative ways of going out into the community and finding these cases and drug-resistant TB cases. Just to summarize, I've told you about programmatically incurable forms of TB, various things we really need to think about doing to prevent amplification of resistance in the first place, and to prevent loss of new drugs. And I mentioned all these factors that are listed here. And perhaps fundamental changes are required as to how we select anti-TB drugs. And finally, I firmly think that active case finding is needed. And if we're going to really address the problem of TB and drug-resistant TB, and of course we need precision diagnostic tools. Otherwise, we're flying blind, and we're really going to have amplification of resistance because we're not really going to know what we're treating. So I want to acknowledge Tawanda Gumbo, and his lab did all the drug levels and collaborated on the study, and Rob Warren, who did all the MIC and culture work, and many people in my lab and the agencies that funded this work. So I thank you for your attention.
Yes. No. So before we do, we'll do any questions? I had thought we'll do those at the end, but if people have a burning question for Professor Dedra here, please step over to the microphone. There are microphones in each.
Is that on? So Kieran, that was really scary, what you just presented. I hadn't seen the data about the MICs in the cavities. It really, it's discouraging because we're not going to know MICs in cavities easily from sputum assessments, as you've shown. Increasing drug doses isn't probably going to do it. But not everyone, I mean, in fact, most people don't develop resistance. And if it's really a question of just drug penetration into cavities, why is it that some people develop resistance in that context and others don't?
Thanks, Megan. I think that's a very good question. I think it's a composite of a lot of things. So firstly, we studied highly drug-resistant individuals. But I think, the first thing to say is that, resistance predicts outcomes, and when we look at fluoroquinolone resistance or resistance to a particular drug, it doesn't very accurately predict outcomes. And maybe, as I said, as clinicians, when we see these patients with either INH monoresistance or MDR-TB, and then they turn out to be XDR-TB, maybe this is what's happening. So I think that's in some patients. I don't know the clinical significance of this. We don't know. Maybe, all these populations in the cavities stay like that, and many of them don't cause clinical problems down the line. We don't know. And exactly what the clinical significance of this finding is, we don't know. So, but I think, I mean, there was a paper I quoted of John Metcalf looking at micro-heteroresistance. We can detect that in sputum using deep sequencing, and I think what remains to be seen is what is the clinical significance of this finding.
Kirtan, when I talk to people about precision medicine for drug-resistant TB, they often tell me that, well, it's possible in North America and it's possible in Western Europe, but it's not possible in settings like Southeast Asia or in South Africa. Is it possible?
Yeah, so I think there are two parts to that. I think it is possible. I mean, already in South Africa, for example, there's several studies ongoing. I think Narey is doing a study looking at impact of sequencing. Nazir Ismail is doing sequencing, and we could certainly do it with the centralized laboratory system we have in South Africa. So that's the first part of it. The question is, what is going to be the clinical impact and how will it improve outcomes in patients? But the second part of it, I think the big hurdle is enabling sequencing directly from sputum, because we know in as little as 2 or 3 weeks you can develop fluoroquinolone resistance. And I think that's the big hurdle, and I think we're far away from that. So I think if we get the technology, we will be able to use it, but I think we're going to have to cross that hurdle, and we're going to need some amplification step of some sort to try and address that question. I think that's going to take a few years.
With the line probe assays and also with the new cartridge from Cepheid, resistance against fluoroquinolones and against second-line injectable drugs can be almost on-site be identified. I think the concerns of the colleagues is not the diagnostic part, but is to give individuals individual drug combinations. And there is a disbelief that that is practically being done in settings where there are a lot of patients.
Yeah, I think it's an evolving area, and I think we've started doing that, and I think as time goes, certainly the capability will be better, but I think the big question is still, what is going to be the impact?
Our next speaker is Professor Narey from Harvard. She is going to talk about more sequencing and is really the leading expert in sequencing of MDR TB and XDR TB and the clinical implications thereof. Megan.
Do I know how to find my... great. Thank you. Okay. Well, I just wanted to thank Kirtan and Twande for inviting me to this. Kirtan also set me up very nicely because he had asked me to talk about the prospect of whole genome sequencing for detecting drug resistance with the question, is it ready for, is it for prime time and for clinical care? So back in 2013, this letter to the editor appeared in the New England Journal of Medicine. It was from a group in England, Claudio Kozer and colleagues, who had used whole genome sequencing on a patient who had been presented with XDR-TB. He was really challenging to treat, and they decided that they could go ahead at the time and sequence his strains. And what they found was that he had several strains, several Beijing strains in his sputum, and that both strains were resistant to an array of different antibiotics. There was some overlap, as you'll see in this graph. But that the drugs, the long list of drugs to which he was resistant by genotype corresponded very closely to those that they predicted by doing drug sensitivity tests. So that was somewhat remarkable because although people had been doing sequencing for years at that point, they hadn't been using it as a clinical tool. So we fast forward to 2017, back in May, when Public Health England announced that they were going to full program whole genome sequencing for drug resistance. So the current strategy in England, when people present with TB, is that they get both a phenotypic DST, but their isolates are sent to a single reference lab where DNA is extracted, and they also get whole genome sequencing. So the important thing to note here is that they're not sequencing directly from sputum, as Kirtan pointed out. They're growing TB usually in midget, and then as soon as there's growth in the culture, it's sent for DNA extraction. And that has been perhaps not a scientific breakthrough, but a really important public health breakthrough to make something like sequencing just standard routine care. But England is a low burden and high income country, and that doesn't mean that everyone else can do this. One way to think about this is to ask the question whether what we've already come to expect based on using GeneXpert and the Hain test, whether we, and the target product profiles that WHO has produced around drug resistance testing, whether that's going to be met by the kind of sequencing that's currently being done. And if you take a look at my really, a very brief summary of the things on the target product profile, very few of them are actually met. So yes, in principle, you could cover a lot of drugs, and we'll come back to that later as to whether we're covering all the drugs and whether we're getting good sensitivity and specificity for all the drugs. In terms of its ability for temperature and humidity, I mean, we're using big sequencing machines. We'll come back to that as well. So probably not. And if you go on down the list, you'll see that almost all the things that we expect from GeneXpert, we expect from new diagnostics, are not yet there for whole genome sequencing. So why would we do it? Well, one thing about the current tools is that although they pick up the majority of resistance to RIF and INH, they pick up less resistance to second-line drugs. And there's a long tail of rare mutations that probably explain between 5, 10, maybe even 20% of resistance to some of the more second- and third-line drugs that we use. And we're never actually going to capture them unless we have a tool that picks up all those mutations. And at the present time, these other tools don't do so. So are we there yet? Well, it depends on where we think we're going. If we're going for use of whole genome sequencing as a point of care or near point of care tool that competes with current technologies in high burden settings, probably not. What we really want is to skip that culture step. We want to have people produce sputum, and I've got little DNAs coming out of that person's in their sputum, go directly into a tool that produces a sequence, have that sequence go directly into a tool that produces a prediction about drug resistance, and have that prediction be pretty accurate. So what are the steps that would have to be accomplished to get there? Well, actually, there's been 3 studies that have come out in the last couple of years that have done this already, that have taken sputum and produced a whole genome sequencing with the idea that it would be used for drug resistance predictions. The first study came out in 2015, and actually it was able to use whole genome sequencing directly on sputum samples to identify TB, but there wasn't enough TB DNA in the sample to actually do the drug resistance predictions because there's lots more mutations that they would have to detect. In a second study produced last year, Brown et al. Used a different technology, something called SureSelect, which we'll come back to in a moment, and got better coverage. So they were able to do drug resistance predictions on most of their samples, but at very high cost and a fairly slow turnaround. And then in another really, I think, breakthrough this year, the British team showed that you could do same-day sampling directly from sputum and get whole genome sequences that allowed the prediction of drug resistance in about 20 out of 27 strains. So not perfect, but well on its way to being a usable tool. Turnaround time was between 44 and 16 hours, depending on what type of sequencing was used, and the cost was either around £100 or a little bit more, again, depending on the sequencing technology. Okay, so what are those steps, and can they be further improved from that? Well, they're the obvious ones. We still have to collect sputum. We have to liquefy it and decontaminate it. And then we have to get DNA out of the TB. It's in the sputum. We have to enrich it, which I'll come back to in a moment. We have to amplify it, sequence it, call the variants, meaning the mutations, analyze whether those mutations actually are the ones that are associated with resistance, and really importantly and very neglectedly, get the results to a clinician who can use them to make a practical and fairly immediate choice about how to treat a patient. So we know how to do the first step. We have lots of experience collecting sputum, liquefying, decontaminating. We know how to do this many different ways, so we'll skip that. The problem with TB is that it has this horrible waxy coat that makes it relatively impenetrable to all the drugs for which we treat other organisms, but not TB, penicillin, etc. But it also means that it's hard to get things out, including DNA. So when many, many studies have shown, and this is the one I put up here is just an example of one of them, that using the sort of standard technologies that we use to get DNA out of pathogens or cells is not very high yield. In fact, the best we can do is a yield of around 60%. So we actually need to get all the TB DNA out of those sputum samples that we can, because we're competing, or the TB bacilli is competing with a lot of other cells, human cells and other flora, and we don't want those in our sequencing specimen. So the graph on the bottom there just shows the difference in the yield between a direct sputum sample and a midget sample in the study I just cited from Britain of this year that showed that you could do direct sequencing. And you see that cultures obviously do much better. We grow up to much higher yield. And in about a third of the cases, there was not enough DNA to reliably do whole genome sequencing using our current extraction strategies. So those need to be improved. So not only do we have this waxy cell wall, but we have all these cells. The more polys or neutrophils are in a person's sputum, that's more human cells. That means there's going to be a lot of human DNA that is extracted when we extract the TB bacilli's DNA. There's also other nasopharyngeal flora and other flora that's just there for other reasons. And we have to try to just pull often the needle out of the haystack of other DNA, the needle being the TB DNA that we want to sequence. So how do we do that? Well, this is, again, from the same paper showing, if you look across the x-axis, those are a number of different patients who have different amounts of different kinds of DNA when on sequencing. And what you see is for these direct samples, many of them have a lot of human DNA, and many of them have a lot of nasopharyngeal and others. A few are mostly TB, but that's unfortunately only a few. So this is what we are contending with, and it's not something that we contend with when we grow TB up in culture. So how can we improve that? Well, there's two general approaches. One is to try to get rid of human DNA, and the other is to try to select for TB DNA. And both of those have been done, and sometimes they've been done together. So all of those studies I've mentioned have used one technique or another to try to improve the enrichment of TB DNA. A standard approach is to use enzymes that are specific to humans to lyse human cells, wash them out, and then get rid of what's there and leaving TB DNA and other pathogens intact, and then breaking down their cell wall and getting the DNA out. Other approaches are to selectively bind pathogen DNA, which tends to have non-methylated CpG groups, or to use detergents that are specific to either the pathogen or the host to selectively get rid of human DNA and leave TB DNA. The other approach is to go out and try to capture TB DNA, and there's 3 or 4 ways that one can do that. One can hybridize it by taking the entire genome, breaking it up into little bits and sticking it on a microarray, capturing the TB DNA through hybridization, and then sequencing it. And that's an approach that the second group that I talked about used. It's a commercial product that Agilent makes called SureSelect. They will produce for you a tiled genome on a chip at a cost, and the cost is quite considerable. So, so far, that hasn't been a way that we are considering for sort of routine diagnostics. Another way is to use something called molecular inversion probes, which are a kind of a circularized probe that hybridizes at either end to the DNA target. And then when the either end hybridizes, then they come together, the probe forms a circle. That circle can then be cut with an enzyme, and then trimmed, and then those fragments can be sequenced on a next-gen sequencing tool, as if they were just fragments of normal fragments of randomly broken up TB DNA. And that gives you very high coverage, meaning the issue that we've been talking about, heterogeneity, is actually dealt with because you can get 1,000-fold coverage, meaning that you can go very deep into the sample of what's there. And if there are rare variants in your sample, you can pick them up. And then there's the other approach that Kirtan mentioned, which is to use PCR, and instead of PCRing at a single target, PCR all targets, and put all those amplicons into a sequencing tool and then sequence them. Technically very challenging because of interactions between PCR product and between this intense multiplexing, but it has been done, not for the whole genome, but certainly for the drug-resistant mutations, or genes. So then we come to what kind of sequencing do we want to do? So the study that I mentioned sequenced on several different sequencers. Most of them were Illumina next-gen sequencing, which is sort of standard approach to how to sequence at the moment. Those come in 3 brands: High-seq, which is cheaper, but batches things together and does very high throughput, but it takes a little bit longer. A smaller version, which is called MySeq, which a lot of scientists have in their lab and could be put at the level of a sputum smear microscopy lab. And then a Mini-seq, which is even smaller and more handy and prevents this problem with batching. But each of them has their own price. The High-seq is obviously cheaper per sample, but much more expensive for the machine. And they take various amounts of time. So a High-seq large batch might take up to 2 weeks. The alternative that Kirtan also mentioned is something that is just beginning to get a lot of attention. This is Oxford Nanopore's Minion, which is a tiny little sequencer that uses a different technology, a different chemistry to sequence, and has the capacity to be a point-of-care sequencer. So, in fact, it was used during the Ebola epidemic to sequence Ebola directly from blood samples of Ebola patients, but really only 1 or 2. And it's actually a great tool. It's much cheaper than other forms of sequencers, $1,000 for the sequencer, but it needs a cartridge, and you can only do 12 samples per cartridge. Each cartridge is $500, so the cost isn't entirely clear at this point. Once you have sequence, however you get it, from Illumina, from PacBio, from Minion in the future, it has to go through a bioinformatic pipeline. Unfortunately, these have been really mastered. So what that means is the sequence has to be aligned. There are little bits of TB DNA that have to be lined up to a known sequence. And then someone has to, or the algorithm has to pick out what is different between the reference strain and the strains that you're looking at. And then do something called variant calling, which means where are the mutations, what kinds of mutations are they, how confident in those mutations are you based on the depth of coverage you get from sequencing. Now, fortunately, that process has been really automated, and there's a number of tools, I'm going to just go back for a second, that are publicly available that will produce a result of variant calls very quickly and with almost no user input. And we'll come back to those in a moment. One of the problems with the sequencing tools that we've mentioned is they all have their own error rate. And if you think about how rare mutations are, there's 4 million base pairs of nucleotides in the TB genome, and we're talking about picking out a couple of mutations that might cause resistance. So 4 million base pairs, your error rate can be pretty low, and you still won't know if the one mutation you see is an error or if it's real. So we really need to look for tools that have low error rates. Illumina has the lowest error rate right now at 0.8%, PacBio at 12%. And the problem with Minion at the moment is that its error rate is still high, ranging from somewhere between 5 and up to 30%. It's going down, and I expect it to go down further. But this has been a major sticking point. If you're having a 30% error rate, how will you know if your mutation is an error or actually a drug resistance call? So as I said, there's these online variant calling and analysis tools. There's a whole slew of them. They're all really good. I'm going to show you one, which is a microbe predictor, which also comes out of Public Health England. You can feed in raw DNA sequence and get out predictions on resistance. And all these other tools are equally good, and they do the same kind of thing. So you can take your pick. These are easy to use in general. They will get easier to use the more people use them. And lots of these groups are asking for user feedback. The problem is that you actually have to know what mutations cause resistance in order to use any of this technology. And we do know a lot about what mutations cause resistance for the first-line drugs. We know a little bit about that for the fluoroquinolones, et cetera. But depending on where you look, at least in some of the studies, and ours are consistent with some of the minimum values here, for some drugs, we can't identify mutations in 60% of the drug-resistant strains, or 40%. So there's only a 60% sensitivity. Similarly, specificity is low. And again, that's referring back to that long tail of rare but frequent enough mutations that they accumulate, and if we don't know what they are, we can't look for them in the genome. So really what that gets back to is developing the knowledge base to make these tools usable once we have the technology to make them easy and cheap. This is just an example of how hard that task is. It's something we've been working on for years, and it's super challenging. This is DST discrepancies between two supranational reference labs that we work with. And you can see, this is unpublished, it just comes from our data. But you can see that even these really good labs don't get the same results on the same strains. We all know that drug sensitivity testing is super demanding, and that slight changes in media and circumstances will give you different results. But we're basing our discovery of new mutations with DST as a gold standard, and it's not a great gold standard. So we have to really think that very carefully. There's a number of ongoing efforts to put together vast archives of DSTs, phenotyped strains, and whole genome sequences to do this kind of work using approaches like genome-wide association studies, the kinds that have been used to look for genetic determinants of human features. And they've been very successful so far, and they will continue to be so, I believe. But again, DST is pretty much the critical junction here, because DST is quite unreliable for some drugs. So in conclusion, yes, whole genome sequencing is not only ready for clinical use, it's being used clinically, it's being used in a national program, large scale, and people are forging ahead with making it work. There's still a lot of technical optimization to do of all those steps I mentioned, but it is proceeding, and people are trying different things, and I'm sure that as time goes on, we will get it better and better. But the success depends on a comprehensive knowledge of resistance mutations, and that requires not only further evaluation of what we know, but ongoing surveillance as we introduce new drugs and we develop new resistance patterns that we don't know anything about. So thank you. Questions? There's very bright lights up here. Questions from the panel?
Megan, that was a great talk. The question is, do you think that we ever going to get rid of phenotypic DST? Yesterday in the FIND Symposium, I know you weren't here, but we heard about a concept of a genotypic biosignature portending susceptibility or sensitivity, and then another biosignature portending resistance. So you know, I know you mentioned for different drugs, we're not quite there yet, but do you think we're always going to need DST? It's an important clinical question because at the moment with the line probe assays, for example, in South Africa, they do a line probe assay on the culture isolate, and if they don't detect fluoroquinolone aminoglycoside resistance, right now, we're not doing phenotypic DST. So we're missing a bunch of pre-XDR and XDRs happening right now in clinical practice. So just wanted your take on that.
Yeah, well, I think there's no point in investing as much as we are investing in these technologies if we're going to continue to do phenotypic DST. I mean, it saves time, for sure, but the cost of doing whole genome sequencing and phenotypic DST for, say, 12 drugs is never going to work economically. I do think that whole genome sequencing and targeted sequencing will give us enough information to treat patients as well as we do now. I think the challenge is not losing the ability to do DST. So I'm not sure we all need to be doing DST at every level, or even the levels that are being done now. But it's going to be important that we continue to monitor for new drug resistance mutations to the old drugs as well as new mutations to the new drugs. And that requires a high level of sophistication around DST, which is hard to obtain. So I can't see it going away, but I think it will be reserved for really specialized labs. That's my hope, anyway.
Thank you. All right, so I want to introduce our next speaker, and that's Professor Tawanda Gumbo from Baylor in Dallas, famous for his work on the hollow fiber model, and he's going to talk to us today on therapeutic drug monitoring in clinical practice for drug-sensitive and drug-resistant TB, when, where, and how. Tawanda.
So I usually start off with this. You can go. Let me move to the next slide. I usually start with this slide here, which shows our current practice at how we dose patients. You know, I look like this sumo wrestler here in stature, and we all give the same doses to all these patients in our TB programs. And what's usually very interesting when you talk to pediatricians whose patients have different weights, they already know that what we are doing in adult medicine is crazy because their own patients have different weights and they always take that into consideration. So it's already in clinical practice. But in TB programs, for some reason, we like this type of uniformity. So why is this important? Why am I talking about this? So human pharmacokinetics, we all learn about them. But in fact, one of the most important aspects of human pharmacokinetics is how variable they are, right? There is a wide between patient variability in all aspects of drug metabolism, drug absorption. So for all, you can safely say, for 7 billion people on Earth, you have 7 billion different concentration time profiles of a drug. That is the force of evolution. I don't know what I did, but this comes from evolution and dispersion of mankind, and it gives strength to the species. But pharmacokinetic variability is part of that metabolism that changes between patients. So each patient should be considered unique to themselves, and they have a pharmacokinetic profile that's unique to them. And there's no average patient. There's no average human. It doesn't exist. We're all different from each other. So what is needed is to identify, is to work backwards and say, what concentrations will need to be achieved in each patient to achieve efficacy? Knowing those differences will always be there. So this is a study here that we published a few years ago with Professor Makileron from Cape Town, where we looked at 142 patients with tuberculosis who had blood concentration drawn at 8 weeks of therapy and at a number of time points. And then what we did is these patients were followed initially for 2 months sputum conversion and then up to 2 years to see if they had relapse or died or anything else related to their TB treatment. And we defined poor outcomes as therapy failure, relapse, or death. So here is just a picture, a snapshot of the pharmacokinetic variability in these patients for rifampin peak concentration there and the rifampin AUC. As you can see, it's a whole range. All these patients got the same dose in mg per kg, by the way. And same with isoniazid, same with pyrazinamide. So what we then did is we used machine learning-based algorithms where we put in all clinical factors, all clinical radiological factors in the patient, including drug concentrations, and then said, what is it among all those factors that will predict outcome? Poor long-term outcomes, good long-term outcomes. And these machine learning algorithms rank the factors by importance. And the most important aspect was pyrazinamide AUC, so the pyrazinamide area under the concentration time curve. And it said if your AUC was less than this, you were more likely to fail. And underneath that, the next factor was rifampin AUC. And underneath that, the third factor was isoniazid AUC. None of the other patient factors and radiological conditions and so forth even featured in the top 5 predictors. They were all concentration related. So we did a similar study with Soumya Swaminathan in India in children. And what you have here is 161 children who were started on treatment. And this slide here describes the characteristics in the children and the clinical outcomes. So we used one of those machine learning algorithms again in the children to see what drove outcomes. And this slide here just shows you the variability in the different concentrations up here on the upper left. This is peak, this is AUC, and time to peak concentrations, different aspects of drug concentration time profiles. There was variability in the children. We should always expect it. What were the most important predictors of outcome in the children? Drug concentrations. Drug concentration. Pyrazinamide, followed by rifampin, followed by isoniazid. Same story. Now, here's what's interesting. In this particular study, when we did this, we tricked the machines. We put in a falsely negative, poor correlation sample and another sample that had 100% correlation with outcomes. And the machines ignored the one that we had put in as a decoy and still went on to pick drug concentrations as the most important aspect. Now, isoniazid did a very strange thing that you'll see at the bottom, which was that high isoniazid peak concentrations were associated with poor outcome. And in fact, what was happening is there was an interaction between rifampin and isoniazid. And essentially, what that shows you is there is a potential negative interaction, there's potential antagonism. So, what does this matter? So if you then said we want to look at death as a hard outcome in these children, and what drives death? So you separate therapy failure and other things, and you just want to look at death in these children. The most important drivers, drug concentrations. And the antagonism is highlighted again. Children actually died more because of this concentration-dependent antagonism. So this is real, this is there, and it always comes up. So now when you look at the drugs that we use in multidrug-resistant TB, this is the WHO treatment guideline. We categorize them into these 4 groups that you have here. And essentially, you pick 1 from each of these groups, 2 from this group C, in order to make your regimen, and somebody else is going to talk about this later on. But I just wanted to put up this slide to key you in to this group B and those drugs to look at what happens with these drugs. So in the treatment of multidrug-resistant TB, field studies show about 52% cure rate. And the group B and C drugs that you saw there have a narrow therapeutic window. And aminoglycosides are related with ototoxicity in 45 to 70%. And by the way, this deafness that the children and adults get is irreversible. So it's not going to go away when you start therapy. And with cycloserine, severe neuropsychiatric symptoms. So we investigated this in patients in Botswana with multidrug-resistant tuberculosis. And essentially, we looked at amikacin, and what you see there is the pharmacokinetic variability of amikacin in patients in Botswana. Peak trough concentrations, AUCs. And the first thing we looked at, so what predicts hearing loss in these patients? Unfortunately, what predicts hearing loss is the cumulative concentration. So these concentrations, of course, are what drive toxicity as well. So they drive efficacy, they drive toxicity. And what you see here, the CAUC is just cumulative AUC. And then lower down, weight was another predictor. But the top 2 predictors were all drug concentrations. So, in fact, this enabled us to draw a relationship between the cumulative AUC and the probability of deafness in multidrug-resistant TB patients. So, this relationship, since I had never heard of it, I actually didn't know there were animal models of ototoxicity. But some smart people had already figured this out in the guinea pig model more than a decade ago, and they had found exactly the same relationships between cumulative AUC and ototoxicity in the guinea pig model, in the albino guinea pig model. And in fact, the factors that we use currently to monitor the peak and trough concentrations were not associated with outcome, with ototoxicity, in that guinea pig study or in our patients in Botswana. We also then use the same technique to identify to identify factors associated with outcome with sputum conversion in the same patients. And unfortunately, the same drug concentrations were associated with outcome. So high drug concentrations were good for outcome, but there were also the same aspects that led to more toxicity in these patients. So this is an example of a narrow therapeutic window, and our second-line drugs tend to have this narrow therapeutic window. So I put in this slide. PK variability happens every day, and it's what's driving outcome in your patients today, yesterday, and next year. Just because we don't measure it does not mean it's not driving outcome. It's always there. It's as certain as the sun rising tomorrow. So, for every TB drug that we have done studies so far, and we've done more than a dozen probably, there's always considerable PK variability. And in each of those studies, those concentrations drive outcome, right? Because MIC, the minimum inhibitance concentration, also drives outcome, you essentially have an interaction of drug concentrations and resistance phenotype, or MIC, that also drives your outcome, which makes it very difficult to guess without checking, without testing. And this is this comes from our simulations on if you looked at proportion of patients who achieve the concentrations that we know drive outcome versus the MIC. And essentially what you see, what this attempts to show is it depends on the dose, but there is a concentration, there is an MIC above which patients start failing therapy. This is from Monte Carlo simulations. In actual patients, this is the study by Aman Van Doorn and group in Bangladesh. And what they have here, what you have here are MICs of gatifloxacin in their very, very well-known study that led to the Bangladeshi regimen. And they also sequenced the gyrase gene here. And these patients had mutations. What do we see? In 161 patients, as your MIC increased, your percentage of patients who succeed starts falling off. And you eventually get to an MIC above which no patient responds. So, I'm going to finish off by saying we need to aim. So the whole point of therapeutic drug monitoring is to allow us to aim, get a good concentration, right? And so, now, if you do that, how expensive is it going to be? So we did a study. We used, in conjunction with the Western Cape Department of Health, where we looked at the number of patients in the Western Cape who get tuberculosis, the proportion that is MDR-TB, and then said, how much would it cost and what would be the returns if we measured drug concentrations in every patient, even one with drug-susceptible TB, and then responded to them by changing our dose using a Bayesian approach. And this, what we would do then, we would do pharmacokinetic sampling at these 3 time points, and then change doses. So we did that, and what we found was, I'll just move on. What we found was, if you looked at these 4, 5, 6 different outcomes, 2-month sputum conversion, death, cure, relapse, acquired drug resistance, and you followed it through, what would happen? And the answer was, here are your gains with that sort of approach. 98% cure, and as you go along, you will see very few patients fail, very few patients have relapse, compared to the current way we do, the current standard way. And in fact, Jotam, who is here, then did a cost-effectiveness analysis, and essentially, within 1.5 years, the health system would have regained its costs if we measured drug concentrations and did therapeutic drug monitoring in everyone, all right? So you frequently hear that this would be expensive. In fact, I would like to argue that it would be very cost-effective wherever it's done. And in fact, if you had your assay costs fall to less than $18 a day, you would realize the gains back sooner and faster. And this just gives, for the Western Cape province of South Africa, the gains that you would get by following such an approach. So I'm finishing. So who should get therapeutic drug monitoring? I would say all MDR-TB patients, especially the ones on aminoglycosides and on linezolid. And soon we've been working out on looking at cycloserine in Cape Town. So we should. Why should we? Why did I pick those? Look at this. So if you have 45% to 70% ototoxicity, which is irreversible, including in children at language acquisition stage, right? And then you said, what's the cost of a cochlear transplant, right? So it's around between $22,000 and $32,000. This is actually just the cost of implantation, not follow-up and all the other things that are required. And then my question is it really ethical not to monitor concentrations since we know the concentrations, the cumulative concentrations associated with the ototoxicity? And we can stop before we cause this irreversible problem. So what is the cost in dollars, and what is the cost in terms of quality of life? In drug-susceptible tuberculosis patients, and I'm finishing, there are some patient groups that are at high risk of failure based on epidemiology and many aspects that we know up to now. Patients with diabetes, with HIV, patients with high bacillary load, and I'll show you a model we have been working on where you can pick out the trajectory of failure in sputum using TTP midgets early, and then local and patient MIC factors should drive each program to determine in whom else to do therapeutic drug monitoring. And this is what I was talking about. We actually use TTPs that we converted, and it turns out that you can actually cluster patients into those that will fail during the first 2 months, into those that will relapse, including those that will respond slowly. And this is the cluster I am showing here. So there are innovative ways using what we already have to pick out patients who would need therapeutic drug monitoring. And this is my last slide. And then there are local factors. So if you have high rate of fast acetylators in your population, and we have shown using meta-analysis that fast acetylators are more likely to fail therapy and acquire drug resistance. Or if you're in a place like South Africa where you have a high allele frequency of SNPs, this drives rifampin, low rifampin concentrations. So your local factors, or if your MICs are high where you live, should give you additional indications in whom to do therapeutic drug monitoring. This is my group here. These people in the lab, we usually boast that is better than the United Nations in appearance. These young people who some are here who are here in the crowd, he does a lot of modeling, Shashi, hollow fibers. Jotam does machine learning, and Devyani does a lot of the hollow fiber work. And I'll stop here.
Right. Any questions for Tawanda?
So I was struck by I think it was the second to last slide where you showed the high rates of hepatotoxicity in INH and RIF. I'm assuming that's in South Africa.
No. So the high rates of hepatotoxicity are actually in China.
Oh.
Yes.
So there's a lot of discussion about being more aggressive about treating latent TB infection in places in high burden settings. There's a lot of discussion about being more aggressive about treating latent TB infections with the hope of really having an impact on TB reduction. What's your sense of the trade-off in those contexts where you think that there is hepatotoxicity from these drugs, but we would be giving them to more people who with less serious disease? Do you have a sense of what the trade-off there is?
Yes, so we did a limited exercise, but we used the South African population and said what are so if you had latent TB and we just looked at isoniazid, and this was a modeling exercise, and I think some people will talk about latent TB later. What are the trade-offs? Assuming the acetylator phenotype in South Africa. So paradoxically, where you have the highest rate of fast acetylators, your concentration-driven toxicity will be low, right? Because you are moving it. So in places like South Africa, there isn't as much of a trade-off in terms of hepatotoxicity. There are other types of trade-offs. In places where where you have slow acetylators and many other environmental reasons for hepatotoxicity, I would say you still want to do your prophylaxis, but monitor. Monitor for the hepatotoxicity.
Okay, great stuff. Thanks. All righty. Oh, all right. Okay, so one very quick question then.
Very nice talk, Dr. Gumbo. I just had a comment, and maybe your thought on this is, you stressed on therapeutic drug monitoring for MDR patients with TB, but what do you think, especially in the pediatric population, where you're putting patients on aminoglycosides and with the high rates of ototoxicity, isn't it more prudent to do therapeutic drug monitoring on them, especially because it's so difficult to get audiograms or audiometry in this patient population? It's relatively simpler to get a blood draw sample compared to doing an audiometry. So is there any effort from the those who make the policy to ensure that we at least do it in this age group where ototoxicity has such significant implications?
Yeah, so very little is, it's a lot of it is unknown in children. But I would actually say that we would just use the same principles we use in adults for now. But very little is known, and how much blood draw and how much you can do, would still need to be worked out. So I'm afraid I can't be definitive on that.
Okay, thank you.
Okay, I just want to ask a question based on when you... Oh, sorry.
Yeah, I think we won't take any more questions. We're way behind time, so you can speak to Tawanda at the end.
Okay, thank you.
All right, sorry.
I'll do that. It's fine.
Okay, so we're going to move on to our next talk now, and in fact, there's a parallel session going on now where they're reporting the preliminary results of the Stream 1 study. And so short course WHO regimen and use of new drugs quite controversial. So to address us on this is Professor Christoph Lange from Borstel in Germany, and his talk is entitled Key Practice Points, Short Course WHO Regimen and the Use of New Drugs like Bedaquiline and Delaminate. Welcome, Christoph.
So thank you very much for coming here, and thank you also very much to the Lancet Respiratory Medicine for providing a forum for tuberculosis in the last few years. I think the Lancet journals have been very gracious for the tuberculosis scene to promote TB research at the front line, and it's one of the most prestigious journals that we have in the field. So thank you very much for also supporting the symposium. So I have two topics. The one is the shorter course WHO regimen, and I will start with this, and the other one is the use of novel drugs. So what do we know about the duration of therapy? We heard today that probably prediction of drug resistance by genotypic methods, whole genome sequencing, will matter on how well patients are treated. We heard today also that therapeutic drug monitoring will matter to precisely treat patients, and probably the duration of therapy that patients get also will matter, because why should all the patients with MDR-TB get 20 months of therapy? Why do all the patients with TB get 6 months of therapy? This is a historical trial published in 1976 on two different arms with rifampicin and isoniazid high-dose regimens, 1,200 milligram rifampicin and 900 milligram isoniazid daily plus streptomycin versus every second day. And if you look at the relapse rates 1 year after treatment was finished, between 14.9% in arm A with the daily dosing and with the every other daily dosing 11.4%, you see that more than 85% of the patients have actually achieved relapse-free cure with a 3-month regimen. We should have much more emphasis on individualizing the duration of therapy, because patients with TB get so long therapy than almost in no other bacterial infectious disease. And I'm convinced that the 20 months or the proposed 9 months for the short course regimen are wrong for the majority of affected patients. So what is this short course regimen? The short course regimen is derived from the Bangladesh regimen. It's a fixed dose combination of 7 drugs in the beginning: kanamycin, moxifloxacin, proteonomid, clofazimine, pyrazinamide, high dose isoniazid, ethambutol, followed by 5 months of moxifloxacin, clofazimine, pyrazinamide, and ethambutol. This was also tried in Niger and in Cameroon, and in all 3 settings had very high treatment success rates. And this encouraged the World Health Organization to propose this regimen, and they say that the majority of affected patients with MDR-TB would benefit from this. I am not so sure about this. So what are the requirements for having the shorter course regimen? First of all, this is the most important requirement: the confirmed resistance or suspected ineffectiveness to a medicine in the shorter regimen, except to isoniazid, is an exclusion criterion. Then it's exposure to second-line drugs in the past, it's intolerance in pregnancy and extrapulmonary TB, and also if medicine is not available is an exclusion criteria. And also, only if the exclusion criteria are actually excluded, then patients will be eligible for this shorter course regimen. So we looked at the WHO region Europe, where the largest group of identified patients with MDR-TB live today, and asked who is actually eligible for the shorter course regimen in this area. So this was the first study, basically a combination of analysis from different databases, 5 different databases, one from Austria, from France, from Germany, from Portugal, and then from 17 different countries in the TBNet database. Here are 612 patients included where the bacteria had full drug susceptibility testing out of 1,100 patients. So 612 with full DST. And in the end, and only by criterion number 1, only 7.8% were eligible for the shorter course regimen because the bacteria were susceptible against all the drugs in the regimen. In another cohort, also from Europe, Janina Balbanova and colleagues looked at 4 different other sites from Estonia, Latvia, Belarus, and Romania. And they found that only 4.2% of the patients would be eligible for the shorter course regimen because either there was drug susceptibility to one of the drugs or there was previous treatment. But the majority of these patients were actually excluded because not all of the drugs were susceptible in the regimen. If we look in further detail, one of the exclusion criteria is that not all of the drugs should be available in the program. Kanamycin is not available in any of the European Union countries, except from the Netherlands, where in the hospital pharmacy in Groningen, they produce kanamycin for themselves. But apart from this, in none of the EU countries, kanamycin is available. So formally, this would be an exclusion criterion, but everybody would probably agree that it could be exchanged for amikacin or capramycin. Also, if we look at the nature of drug resistance causing isoniazid resistance, you see that there is high-dose isoniazid in the regimen. Then we see that about 90%, and in other studies it's actually 95%, are due to the mutation in the CAG gene on position 315. And this is corresponding to a high-level mutation. We recently looked at 60 different strains by MICs, and none of the strain had a low or intermediate resistance. So only 10% or 5% of the patients in the European region approximately would benefit from high-dose isoniazid. This is different in South Africa, where there is a larger group of patients that do not that have bacteria that do not have the CAG315T mutation. If we look at isoniazid, these are 55% at patients who have isoniazid resistance that is based on resistance in an INHA promoter, either on position 8 or 15 or 16. And this is usually related to a resistance to proteonomide, which is also part of the regimen. So the high-dose isoniazid doesn't really make sense for the patients in this region. Either isoniazid is not working or there is proteonomide resistance, which then would be also one drug that does not work in the shorter course drug regimen. So the key practice points from this are that the shorter regimen proposed by the WHO is not applicable for the majority of patients and actually not applicable for the vast majority of patients in the European region of the WHO. The regimen should not be given to patients as a standard regimen, but only if the results of drug susceptibility testing suggest that the regimen would be working. And also fixed drug combinations and fixed durations of therapy are probably not the solution. We have heard from Megan Murray that we're now able technically to have tailor-made regimens based on the results of molecular drug susceptibility testing and confirmed by phenotypic drug susceptibility testing. And hopefully we will have biomarkers that can guide us in the duration of therapy in the future. So when should newer drugs like bedaquiline be included in a regimen? And I exclude actually delamanid because the knowledge on how well delamanid works in a regimen is still very poor. The only knowledge that we can rely on at present are the 2-month culture conversion data. And we're eagerly awaiting tomorrow the presentation from the Otsuka representatives on the outcomes of the phase 3 study where bedaquiline has been used for the treatment of patients with MDR-TB versus placebo. So in this year, there have been 3, I think, important studies that show the benefit that bedaquiline-based regimens could have on culture conversion in patients with tuberculosis. And we saw this morning or around noontime presentations from the NTB group that even in highly pretreated patients with MNXDR-TB, there's more than 85% of sustained culture conversion on bedaquiline-based regimens. This study was performed by the Blini group around Lorenzo Guglielminetti and showed that there were 96% of patients who had bedaquiline-based regimens achieved sustained culture conversion, low number of 25 patients, and the same was also seen by the group in Borstel, here headed by Johanna Olaru, where all the 20 patients treated on a bedaquiline-based regimen achieved culture conversion within 6 months. And this was also summarized in a multi-center study and a retrospective multi-center study where Ketan was also part of the group, where culture conversion in more than 90% was achieved at the end of treatment in patients with MDR-TB on bedaquiline-based regimen. This is far away from the around 50% of cure that are reported in the latest WHO report on outcomes in MDR-TB. This is basically a honeymoon that we see currently in the outcome of MDR-TB treatment, and we must use these drugs very wisely so that we do not lose it for the treatment of our patients. We can use the drug with treatment algorithms, and this is just one suggestion of a treatment algorithm proposed in the New England Journal. There was recently another one in the European Respiratory Journal by José Caminero. And these algorithms are direly needed because if we want to use whole genome sequencing to predict drug susceptibility, we also need treatment algorithms that we then apply to the results from whole genome sequencing to have automated regimens produced by this information. And this algorithm has 8 steps. And here you see bedaquiline on step number 4. The first step would be to look for rifampicin resistance. If it's a low-level resistance, add rifampicin, but do not count it among the drugs. The same in the second step for isoniazid. If it's a low-level isoniazid resistant, add isoniazid, but don't count it. Then is there a moxiflox or a fluoroquinolone resistance? And if this is present, then bedaquiline should be used. You see that delamanid is here only on step 7, so it's downgraded because the quality of data on bedaquiline is currently better than on delamanid. This may change by tomorrow afternoon, but at present there is not enough data to lift delamanid up to in this algorithm. So this key point number 2 is that DST-guided bedaquiline regimens lead to very high rates of 6 months culture conversion, and we're now very confident from different studies that 6 months culture conversion rates are very good proxies for relapse-free cure. Bedaquiline is the key drug for MDR-TB treatment in patients where the bacteria are resistant to fluoroquinolones. And the role of delamanid for the treatment of patients with MDR-TB is presently less clear. Thank you very much.
Right, so any questions, comments for Christoph? Go ahead.
Thank you for a very good talk. I just want to ask, when you talked about that bedaquiline it might be the best drug now because it showed 90% culture conversion. The first talk that we got from Prof. Kieran, he talked about there's about 38% of bedaquiline resistance which has been shown. So is the mutations or resistance also put in consideration when they do see that bedaquiline could be the best drug to help in MDR-TB resistance?
So currently bedaquiline is on D2. So this is like the second worst group in the hierarchy of TB drugs that is proposed by the WHO, and that is probably not justified. It should be much higher up. How much or how high up it should be, we currently do not know. But certainly drug resistance to bedaquiline can develop. We've seen last year the publication of the same patient in 2 different journals, and that where the bacteria acquired resistance against bedaquiline and delamanid. And I'm sure that all of us will see the development of bedaquiline drug resistance, and that will jeopardize the honeymoon period that we currently see in the treatment outcomes.
All right, so thanks, Christoph. And so now to switch gears slightly and moving on to latent TB infection, our next speaker is Professor Neil Schluger from Columbia. His talk is entitled New Tools for TB Elimination, Treating Latent TB in the 21st Century. Welcome, Neil.
Thanks very much. Thanks, Kirtan. Thanks to all of you for staying. I realize, according to the schedule, my talk's already over, but I'll try to get through this quickly. Sorry. Okay, everyone here is familiar with what I'm showing you here, that the vast majority of people who are exposed to Mycobacterium tuberculosis will never develop disease. Most people will develop a latent state of infection, as you can see here. A small number of people, small percentage of people who are exposed to tuberculosis will develop active disease quite soon after that exposure. That's only about 5% of people. The vast majority of people will develop a state of latent infection. Of this pool of people with latent infection, 5 to 10% will develop reactivation sometime in the course of their life. What that means is that there's an enormous reservoir of people, estimated somewhere between, let's say, 1.7 and 2 billion persons in the world that constitute this reservoir of latent tuberculosis infection, of whom 5 to 10% will generate cases of active tuberculosis at some point in their life. Therein, in a sense, lies the challenge. it's been recognized in several mathematical models that have been published over the past several years that because of this enormous reservoir of latent tuberculosis infection, it's unlikely that tuberculosis can be eliminated without treatment on a fairly large scale of persons with latent tuberculosis infection. And WHO, in this document that was published a couple of years ago, whoops, sorry, I keep pressing the wrong button. In this document that was published a couple of years ago, recognizes this, and in this framework, action framework for elimination of TB in low incidence countries, low burden countries, recognizes that it will take, as I mentioned, widespread treatment of latent tuberculosis infection. Now I think, and Megan alluded to this earlier, I think that many people think that widespread treatment of latent tuberculosis will be an important tool for elimination of tuberculosis in high-burden countries as well. Certainly you can't eliminate any infectious disease without stopping transmission, so treating active cases is extremely important, but you'll still be left with this enormous reservoir of latently infected persons who will need to be treated at some point. So I think there's a growing realization that treatment of latent tuberculosis infection really is going to be a key tactic in tuberculosis elimination around the world. The question is that possible? Is that feasible with the tools we have now? So the challenges, I think, are to doing this are listed on this slide. If you think about using treatment of latent tuberculosis infection as a means to TB elimination, you have to deal with certain realities. The first is that most people with latent tuberculosis infection will never develop active tuberculosis, even if you leave them alone. So that means if you're going to use massive scale treatment of LTBI, you have to accept the fact that you're going to treat a large number of persons who are healthy to begin with, even though they have latent TB, and would never develop that infection, develop active TB under any circumstances. So, as I said, you're going to have to treat a huge number of people, most of whom will never get sick. And so because of that, I think large-scale treatment of latent tuberculosis infection really has to satisfy at least 3 requirements. It has to be convenient for patients. Most people who have diseases, whether it's tuberculosis or diabetes or hypertension or asthma, are not very good at taking medicine, right? Much more so for people who are essentially healthy and feeling well. So whatever treatment we have for latent tuberculosis is going to have to be convenient for patients. Because it will have to be applied on such an enormous scale to be used as a tool in TB elimination, it's going to have to be inexpensive and cost-effective. And then thirdly, again, because most of the persons who are going to be treated are healthy to begin with and would never get disease under any circumstance, never develop active disease under any circumstance, the treatment has to be safe and well tolerated. So I think all those 3 conditions need to be met. It's got to be convenient, it's got to be inexpensive, and it's got to be safe and well tolerated. What does that look like? Well, these are the people with latent tuberculosis at highest risk of developing active tuberculosis disease. And if you were going to start an aggressive treatment program for latent tuberculosis infection, certainly you'd start with close contacts of active cases and HIV-infected persons. But all of these other people on the list are at higher risk of going from latent to active TB as well. People with recently acquired infections, particularly healthcare workers, working in TB hospitals, that's really who's in that group. People who are medically immunocompromised because of medicine they're taking, and other people with chronic illnesses. So these are the kind of people you're going to focus on. What tools do we have? So for the last many, many years, the standard approach to treatment of latent tuberculosis infection has consisted of a prolonged course of isoniazid. The United States Centers for Disease Control and Prevention recommends 9 months of isoniazid for the treatment of latent infection. In many places, the recommendation is 6 months. But I think operationally, the challenges are the same for both regimens. So there's some good things about 9 months of isoniazid. It's extremely effective. We know from studies going back to the 1950s that it reduces the risk of going from latent to active tuberculosis by at least about 80%. Isoniazid has been off patent forever, so it's very inexpensive and therefore universally available. On the other hand, it's 9 months long, and so that's a long course of treatment for people to adhere to. and as a result of that, treatment completion rates, as I'll show you in a moment, are quite poor, and as been alluded to previously, adverse effects are quite common with this regimen. These are just a little data to show you how poorly isoniazid for the treatment of latent tuberculosis infection is accepted by patients. This is a study from North America, published a few years ago. And all I want you to see here is that, right off the bat, almost a fifth of patients to whom isoniazid for treatment of latent infection is recommended, almost a fifth of them decline right off the bat. So that's 17% in this study who have zero adherence to the regimen before you've even started. And then these are data, I think, representative of patient experience probably all over the world. These happen to be data on the left from the New York City Department of Health TB control program. Is a very good TB control program, showing you that adherence completion to a regimen of 9 months of isoniazid is less than 50%, sort of in the 40 to 45% range. So if you start with, let's say, 100 people, and 17 of those people refuse to take the medicine in the first place, and of the remaining 83, fewer than half, let's say 40% or so, complete the regimen, you can see that you've done a very ineffective job of providing latent infection treatment to the population you're seeking to treat. So can we do better? So I briefly want to go through newer regimens for the treatment of latent tuberculosis infection. So a shorter regimen of 3 months of daily isoniazid and rifampin has been used in some parts of the world for many years. I'm not going to say too much about that regimen, but there is quite a bit of attention and interest in regimens. The other regimen shown here, a regimen of 4 months of rifampin given daily, and then a regimen of 3 months of isoniazid and rifapentine, sort of a long-acting rifamycin, given once a week. So that's 12 doses of medicine for the treatment of latent tuberculosis infection. And so what do we know about these regimens and their potential use? So the 3 months once weekly regimen of isoniazid and rifapentine is one that we studied in the Tuberculosis Trials Consortium. This is an international TB research unit funded by the US CDC, and this was a study we did over a long period of time. The results were published a few years ago. Large study, 8,000 patients who were randomized to get 9 months of daily isoniazid, the standard treatment, that's 270 doses of medicine, or 3 months of isoniazid and rifapentine given once a week for 12 weeks, so essentially 12 dosing days of medication. All patients were treated and then followed for 2 years after completion to ascertain the rate of development of active TB, and in the standard therapy arm, isoniazid only, 15 cases of tuberculosis developed in the 2-year follow-up period, and in the 12-dose regimen, only 7 cases of TB developed. So this met to a very significant degree criteria for non-inferiority, so a 12-dose regimen was shown to be equivalent to a 270-dose regimen for treatment of latent tuberculosis infection. Now, the question is, how well tolerated was it? It certainly was effective. Was it well tolerated overall? It was quite well tolerated. If you just take all adverse events without delineating them as to cause, just by grade, there was certainly a trend toward fewer adverse events in the 12-dose arm of this study, and if in particular you look at hepatotoxicity, there was a very clear signal that the 12-dose regimen was quite a bit better tolerated in terms of liver injury than the standard 9-month regimen for isoniazid, and you can see that here. Either, however you look at it, total events, events per patient, events per patient month. So this seemed to be a well-tolerated regimen in terms of hepatotoxicity. But I think there were certain unresolved questions with this regimen when we did this experiment. Those 12 dosing days were all given under a regimen of directly observed therapy. And if you want to treat millions and millions of people for latent TB infection. I think that's impractical from an operational point of view. So must this regimen be given by DOT? The consortium has done a follow-on study, which should be published in the next couple of months, where 1,000 patients were allowed to self-administer, and the good news from that study was that there seemed to be no safety signal in allowing patients to self-administer. The adverse events in that study occurred at a similar rate in the parent study, where all doses were given by DOT, so I think that was reassuring. Adherence was a little bit less, but still quite good. I think an important question is, in the 4-month rifampin regimen, and to a certain degree in our regimen, do these regimens of high doses of intermittent rifamycins lead to the development of rifamycin resistance in people who go on and develop active TB? We didn't really see a signal in this study to indicate that, but it underscores the importance of excluding active TB in anyone treated for latent infection. One major unresolved issue is how useful is this regimen in the treatment of patients with HIV infection. Obviously, particularly in sub-Saharan Africa, co-infection with HIV is not only extremely common, but the major risk factor in moving from latent tuberculosis to active TB. So just to address some of those things, these are data from a network analysis that was published originally in 2015 and just recently updated in the Annals of Internal Medicine, pointing out that these alternative shorter regimens, 3 months of daily isoniazid and rifampin, 4 months of rifampin, 3 months of isoniazid and rifapentine given once a week, the regimen I just described to you, all seem at least as effective or somewhat more effective than a standard 9 or 6 month regimen of isoniazid. And the 4-month rifampin and the 12-dose isoniazid and rifapentine regimens would be predicted to be quite a bit less hepatotoxic, that is to say, quite a bit safer in terms of liver injury than the standard 9 or 6 months of isoniazid. So I think they have clear advantages there. Now, what about this question of their utility in patients with HIV infection? Because, as I said, this is certainly a major issue in treatment of latent infection in sub-Saharan Africa and several other regions in the world. It was shown 25 years ago that by Bill Popp in Haiti that treatment of latent infection, latent TB infection in persons with HIV was effective. These are the data from that study that was published in the Lancet, as I said, nearly 25 years ago. But what's the best strategy for preventing active TB in persons with latent infection and HIV infection? And I'll show you a few studies quickly that really just raise all the important questions that I think we don't yet have answers to. So this was a study of 6 versus 36 months of isoniazid in HIV-infected patients in Botswana, and what it showed was that 36 months, that is essentially continuous isoniazid for 3 years, was associated with a lower rate of development of active TB than just 6 months of isoniazid. Those 3 years was better than 6 months in this study in Botswana. On the other hand, in a study in Brazil, 6 months of treatment of latent infection in patients with HIV infection was associated with long-term protection of development of active tuberculosis. So 6 months in Brazil seemed to work better than 6 months in Botswana. We'll come back to that in a moment. In a study that I'm sure many people are familiar with, published just a couple of years ago, done in gold miners in South Africa, there was no prolonged protection even from 3 years of treatment of latent infection with isoniazid. That is to say, while patients were on the regimen, they seemed to be protected, but as soon as the isoniazid preventive therapy was stopped, the rates of TB began to rise again. The 12-dose regimen that I described to you in the study that we did in the TB Trials Consortium, could that regimen be used in patients with HIV infection? We did include about 400 HIV-infected patients in that study, and the regimen seemed as effective as isoniazid and very well tolerated in those HIV-infected patients, but there's an enormous caveat. That study, our study was done in North America, in Canada, and the United States, very low incidence countries for TB, and all the HIV-infected patients in our study had extremely well-preserved immune function. The mean CD4 positive T-cell count was around 485, so certainly not representative of patients in many parts of the world. So what I would say in summary is for patients, treatment of latent infection HIV-infected patients, that the efficacy of this varies by immune status, the CD4 cell count of the patient, whether or not the patient's on antiretroviral therapy. The benefit, I think, varies by continued risk of TB exposure. So a country like South Africa, which has an enormously high rate of TB, is a different environment than even countries like Brazil, which have much lower rate of active TB, and so a lower chance of re-exposure after completion of therapy. Drug-drug interactions between rifamycins and antiretroviral therapy, I think, in this group of patients are a significant issue. The integrase inhibitor backbone, which is now really the core of more and more HIV treatment around the world, does raise issues when patients are treated with rifamycin. So I think all of these things need to be taken into consideration when one is thinking about treatment of latent infection in patients with HIV infection. I think it really is a very local kind of problem, depending on what kind of ARV is available, what's the background rate of TB in the community, and things like that. And then to sum up overall, in terms of treatment of latent tuberculosis infection in 2017, I think it is true that shorter, safer, and effective regimens for treatment of latent TB are available now. As I said, we have a regimen that you can take on 12 days that's as effective as 270 doses of isoniazid. So that's 12 doses over 3 months. There are several groups, including ours, which are looking at shortening treatment for latent TB infection even to 6 weeks. And so those clinical trials are either ongoing or about to start. We're about to start a trial of 6 weeks of daily rifapentine. So I think treatment for latent TB, as I said, has already been made shorter, safer, and effective. And this allows one to at least think about more widespread treatment of latent tuberculosis infection as a tool towards TB elimination, certainly in low incidence countries. And I think if we can get treatment for latent TB even shorter, even safer, even more well tolerated, we can think about using that on a more widespread scale in high burden countries. I think that's probably still a difficult go right at the moment. But I think this could contribute to lowering the TB incidence in high burden countries. I think the optimal treatment of latent tuberculosis infection in HIV-infected persons remains still to be precisely defined, but it certainly will be affected by location, background incidence of TB, immune status, and the availability and uptake of ARV therapy. So with that, I'll stop, and thanks very much for your attention.
Okay, any questions, comments? Go ahead, Sashi.
Hi. This was a very nice talk. I just have a question. You showed that for isoniazid preventive prophylactic therapy, there was no difference between the DOT and SAT. So you showed that for the isoniazid preventive therapy, there was no difference or no safety issues between the DOT versus SAT. So can the SAT be recommended for the regular TB regimen, because that will free up a lot of resources for other program factors?
Right. So I apologize. First of all, it's extremely difficult to hear up here. I think the question was about self-administered treatment versus directly observed therapy. So let me say in general that this is a big believer in directly observed therapy for people with active TB. I think it's completely unrealistic to think that treatment for latent tuberculosis infection should be given by directly observed therapy. You're talking about an enormous number of people with latent TB infection in the world. Even if you target close contacts in HIV-infected people and people taking immunosuppression, it's still an enormous number of people, and it would overwhelm the capability of any TB control program to require that treatment for latent infection given by DOT. And that's why I think the need for extremely safe regimens for the treatment of latent infection is underscored. As I said, I think our consortium will have data coming out showing that this once-a-week regimen for 12 weeks administered by self-administration, taken by self-administration, is safe, and I think will be acceptable.
Neil, what's your view on bedaquiline and delaminid to treat latent TB infection?
Right. So it's a great question. The question about using bedaquiline and delaminid to treat latent infection. I think, as everyone knows, the optimal treatment for exposure to MDR or XDR TB is a complete black box. I doubt that in the near future that there will be clinical trials using those drugs for treatment of latent infection for a whole host of reasons. I think we don't know now, and I think it'll be a long time before we do know about the efficacy of that.
Yeah. Perhaps this is not such an unrealistic scenario for some parts of the world. If we look at Eastern Europe, Moldova has about 42% of MDR TB of all TB cases, and Belarus about 45%. In this situation, you wouldn't give isoniazid and rifampicin, but you also would not like to give bedaquiline if you want to save this drug for those cases who actually have TB and you don't want to create resistance. So this is a huge dilemma.
Yeah, no, I agree with that. Kazakhstan as well, Belarus, Moldova, places like that, extraordinarily high rates of MDR-TB. Yes, one could imagine doing the experiment, or bedaquiline versus levofloxacin or something like that for MDR-exposed patients. But at the moment, no data.
All right. Well, thanks, and I want to thank all the speakers for fantastic talks, and thank you all for attending.